In this topic, you are going to learn how to identify bacteria in contaminated samples using various DNA regions. To identify bacteria using the DNA, it's important to keep in mind that only specific regions are of importance to distinguish between related samples. Scientists have defined certain small regions in the DNA that are beneficial for identifying a bacterium, as they did in the human genome. Let's take an example, a human biological sample like a hair. DNA from this hair can be isolated and compared with the victim's DNA. The whole human DNA cannot be tested, as that would take a lot of time and money. Therefore, we need to take a small sequence. Keep in mind that 99.7% of human DNA is common among all people. In a real case, a thief was able to collect valuable things from a house in which looked to be a perfect crime. On his way out, he decided to try an apple that was on the table. He took a bite and left the apple. He did not know that with the apple, he left his DNA. And this bite led to identify the thief and get him to court. Let's now get to learn the specific regions in the bacterial genome that are used for the detection and identification of unknown samples. The first region is the 16S rRNA gene sequence. And as you remember from module number 1, the 16S rRNA is part of the bacterial ribosome. The 16S rRNA are regions in the bacterial genome that evolve slowly. The analysis of this region is the best choice for general bacterial species or genus identification. The advantages of using the 16S rRNA are: first, there is a very large public database of the 16S rRNA, and the second one, they could help in the identification and discrimination of the target pathogen from non-target pathogen. The disadvantage of using the 16S rRNA that it does not distinguish the target pathogen from its closest relatives. As an example, Bacillus anthracis and some strains of Bacillus cereus. Another analysis for bacterial identification is the multi-locus sequence typing, MLST. In this analysis, certain regions in the bacterial genome are studied. For example, specifically speaking, housekeeping genes. Thousands of these housekeeping genes are available. However, only less than 10 are of forensic interest. Database for these genes is not available, which make it harder to be used for identification. The highest subtyping resolution is required for precise identification of bacterial isolates. Therefore, highly mutable genomic regions must be used. As an example, variable number tandem repeats, VNTR regions. These VNTR regions are locations where a short nucleotide sequence, like CAT, is repeated multiple times. Coming to the end of this topic, what have you learned? First, the genetic material of a microbe is a powerful tool to identify the agent used as a bioweapon. Second, certain sites in the genome are of forensic importance for identifying the bioagent. These include 16S rRNA, multi-locus sequencing typing, and multiple locus VNTR.